Okay, so I think we can get started. Uh, so hello everybody, and welcome to this new seminar series from the Conexión. The idea is to have a way to share our bioinformatics tools. We were thinking of what would be a nice topic for a seminar series for the Conexión, and I think that there are so many talks out there that maybe it was nice to have something more dedicated to the tools. So the idea is that this is a forum to present them in a very informal way because we all know each other. The presentation is recorded and then it will be available at the YouTube channel of the Conexión. That means that this way also serves as a way to promote the bioinformatics tools afterwards. And the idea is to have this once a month, and we will have two presentations every time, so that at the end of the year we can maybe cover 24 tools provided by the Conexión. You also have the program of the seminars at the webpage. And yeah, I hope that you enjoy this seminar series. And I think that with that, maybe I can start because I will be the first to kick off this initiative. So I'm gonna share my screen and put it in presentation mode. Okay, so right. So I'm going to be talking today about a tool that we are developing in the lab already for quite some time, and it has had different iterations of updates. The tool is PaintOmics, and it's in the framework of integrative analysis of multiomics data, and especially focus on the visualization and interpretation. Um, so, just to put this a little bit in context, because integration of multiomics data is a very broad term, and of course we know when we talk about the multiomics is that we are referring to different type of technologies that allow us to measure the genome, the epigenome, also the transcriptome. This is a perspective that we wrote some time ago. And basically, when you think about all this data and connecting this data, there are like two ways in which you can address the problem. So one way is that you focus on the sample, so you are measuring these components on different individuals, and then somehow you use the multi-layer information to cluster individuals of samples and then to predict a newcomer to which group of this clustering will um, belong. But there is another possibility is that you focus your analysis in the feature and that you try to combine the data to create multi-layered networks, right? So in which you have different type of features, genes, proteins, metabolite, and then you can see how they are connected and they change these connections, these network changes between conditions, right? So these are the two ways usually analyzing the data. In both cases, you may find biomarkers that define the clustering or define the predictions or that are important hubs in these networks. Now, my lab usually is more interested in this part because it's kind of helps us to understand the biology, and this is the framework in which PaintOmics was developed to say something about, the relationship between features, between genes and compounds. And yeah, so this is one problem that we see is that usually all this combined information is very difficult to interpret and to visualize and to interpret together. So we want to provide tools that will help to address this problem. So this is PaintOmics4, which is a tool for the integrative visualization and analysis of multiomics data. And basically what we do is that we are able to process different types of omics measurements from gene expression, metabolomics, proteomics, but also omics that have a regulatory role, of they are kind of region-based, of in principle, pretty much any kind of omics measurement that somehow can be linked to a gene of protein ID of a metabolite ID. What we have, this is something that comes in the latest stage of the analysis. So we have other tools that do and combine analysis together, but this is really like the latest step of interpretation. So basically, you have already your process data, you have your measurements, you have even your differentially abundant features, and this is what you will provide to PaintOmics. And then we have the idea of PaintOmics that it maps all this information on knowledge databases, on pathway databases to be more specific. And we have different databases that we support, KEGG, Reactom, and MatMam, which is database specific, more dedicated to plants. And then basically we can do a number of analyses. We can do joint enrichment analysis. We can look at the relationship between, genes and the regulator of the gene. We have different specific things to also look at regulations of metabolites and then visualize the data on the pathways. Okay. So this is the upload interface from PaintOmics. Basically, what you have here is, that you can input or any kind of omics, and then what you do is that you drag this to the main window. These are the selected omics, and these are the omics that you can look at. Also, if you can choose the organism that you are working with, and this is important for selection in the KEGG and the Reactom database. And if we only support, or not only support, we only maintain a limited number of databases, of organisms, but we can add any organism that you may working with. So if your organism is not in the list that you can see when you click here, then you have to request us to add it. And then, for example, here, let me do, you can select which databases you want to look at. So basically, you have a lot of information of PaintOmics. I'm just saying that. So you have different tutorials, and you can also come here and identify the documentation and the previous versions and example data. So it's a pretty well documented resource. So basically how it works. So you have the selected omics, for example, let's say you have gene expression, metabolomics. Usually these are data that is measured in the same experimental conditions of in the same set of samples, right? So it's designed to let's say that in a way, the observations are common across omics. So here is where you put your data files, and basically, you can also preload it in your personal storage space that I will show you later. So there is two places in which you can have your data uploaded. And so the data structure is actually very simple. These are just tab delimited files. So for example, when you are talking about omics features data, like gene expression of protein data, the only thing that you need is a table in which you have the name of the features, and then the values in different experimental conditions. For example, this could be a time series, so you have 3 time points here. And you have usually, you have to provide your data in log ratio, log fold change format, because this helps later on in the visualization. So this is your measurements data, and You can also, usually that's what you do, you can add a list of significant features that you have obtained for whatever the method that you obtained. So this is when you have gene expression of metabolomics of protein data, that's the format. And in the case that you want to input chromatin data, that refers to chromatin regions. Then what you provide is the chromosome position and the start and end of the region, and then some measurement of the intensity of the signal in that region, whatever it is, right? And also, if you have some way, I mean, that's not compulsory, but if you have some way to identify regions that are especially relevant to you, then you also add a file in which you have the indications. But it's important that it's not only significant elements, but that you actually have the full data set, because that's how you can calculate the enrichments per omics type. So when you input region-based omics data, there are some additional information to include here. This is usually, for example, ChIP-seq of ATAC-seq of DNA-seq data. So you will choose the name, your bed quantification. This is the file that I showed to you before. You include it here. You also need to provide the GTF of the organism. We have already for some organisms, we have already a GTF information, and this is, you have to provide this because there will be a way in which we can translate this region-based information to the proximity to genes, let's say. And for that, we need the annotation, so the annotation of the genome. Again, some species are available, but if not, if it's not there, you have to provide yours. And then these are some criteria that you can read more on the paper that we use to assign the region to the neighboring gene, right? So, I mean, that's not... there are more than one way to do that, right? For example, how you define a TSS, whether your TSS is going to be this amount of nucleotides upstream so how you define the region of the TSS, how do you define the region of the promoter, all these kind of things you have to provide. There are, of course, some default values that usually will work for many of you. So if you want to, we also have some way of include information of what we see regulatory omics. And this is in the case that you already have some data that you know that provides regulatory roles. This is especially relevant for transcription factors and for microRNAs, right? So again, this information of which is the potential relationships, I would say, of the already approved relationship between a transcription factor and the targeted genes. So you have to provide this here in this association file, and the same for potential associations between a microRNA and the target gene in this association file. So if you already have this relationship established, then you can say, well, these are my relevant regulators, and that's it. And if not, we will compute some kind of basic correlation between gene expression and this regulatory element within PaintOmics, and then you can modify the parameters of how to compute this correlation in this menu, right? But the idea is that we can also say something about, the regulations within the framework of the pathways. Okay, so once you have uploaded your data, you can browse your data in this storage space. So you can, or you click here and you upload your data here, or in the menus that I showed you before. But in any case, all your data files and also your jobs will be kept in this storage space, of course, up to a maximum, because we cannot have like a so you have a maximum of a space that you can use. You can request for more, but there will be a maximum. So once you have, again, selected your data in the selected omics, right? So this is actually how you start with running. So you have your gene expression, your significant genes, metabolites, proteins. Basically, you click on Run PaintOmics and it starts running, okay? So what happens is that after everything is loaded, and the program will map all the information that you have provided into the pathway databases that we have available, right? So there will be a kind of a huge mapping process. So you can download in a textual format this map data, in which you will have the information, okay, these genes, this region mapped to this gene and this pathway. So you can download this or visualize this with PaintOmics, which is, of course, what you want to do. And here you have already some statistics to see, how well you are matching your gene expression data. For example, in this example, 89% of the genes that you provided were matched to pathways, or in this case, for example, all the proteomics data, we were able to match it to a pathway, right? This is for KEK, Reactome. Usually the coverage is not that good because it has not as many elements. It's a more curated database. So the numbers will be different from one to another database. But you can already have some feeling on that. This is another way to see this. So how is the number of, for each of the omics, which is the number of mapped features and mapped, and which is the distribution of your values, these log fold change values that we indicated, so that you can see that if you are being successful to map your data into the pathways of the knowledge bases. One thing that is interesting is that we have this matching also of compounds. This is for metabolomics data. We don't do any kind of processing of identification of metabolites from, let's say, the raw metabolomics data, but you just provide the names. But what we have seen many times is that there is a lot of ambiguity of how compounds are described, of how compounds are named by facilities and how they are represented in the databases. So we have a kind of text mining strategy that we implemented, some kind of elaborated parsing strategy in which we strongly increase the ability to map any data sets to the information that is in KEK or in the pathways in general, right? So you can browse this here, and if you don't have too many metabolites, you can maybe check if they are okay, or click or unclick some of the possibilities and decide in a more custom way how to do that. But usually this works pretty well. So once you have this, and then you can see. So what PaintOmics is doing is to map into pathways, and then based on the significant features of the relevant features is also compare some kind of enrichment, pathway enrichment analysis is performed. So you have an overrepresentation of significant features in those pathways. And this information can be given here. So you can see how many pathways were found for KEK, Reactome, and how many of them were significant because of your data. Next, this is for cake. You have a little bit more detail in these pathways, how they distribute across the different cake categories, and then we are able to create already some kind of pathway network in which basically you can see how these are connected on the base of shared features that are significant. So you can already start understanding a little bit how this information, your omics information is related to pathways. And then the funny thing the nice thing about this is that you can visualize these networks per omics type that you may want to, right? So you can just use the KEK classification and color based on the KEK classification, but you can color on the base of the gene expression patterns, proteomics patterns, microRNA patterns, etc., that are mapped to the pathways. And you can kind of also adjust the settings on how to see this. But basically what we do is we create with this expression data, we can create like clusters of pathways and then group them together and color them together so that you can see which pathways are changing their expression in a significant way with the same pattern. For example, this is some data from B cell differentiation time course, and this is the data that we have at the gene expression level, and what we can see here is that we have two major patterns. Pathways that are related to metabolism and genetic information processing are part of these first clusters that tend to be downregulated in time during the differentiation process because of the gene expression. So this is how the gene expression behaves, while all these other pathways that are related to signaling increase the expression across the differentiation course. So this is a kind of very visual and very nice representation of this. And this is for the gene expression, but actually, if we look at this was ATAC-seq data, if I believe, or DNA-seq data, you see that the pattern is a little bit different, so it's not exactly the same. There is an increase in metabolism regions being like more open, which is corresponding to the changes in gene expression. But for example, all these genetic information processing is tend to be closed, so because this correspond to this other pathway. And then we have like a kind of breakpoint half of the differentiation process. You have to see how you interpret that, but you can already see that there are some commonalities and some differences and how pathway behaves depending whether you look at the gene expression or you look at the chromatin. And this gives a kind of general overview. So this is the pathway network kind of visual aid. We also compute enrichments, as I mentioned. So what you have is a table in which you have all the different pathways, where there are KEK, of Reactome, of MapMan, and then you have like enrichment values for each of the omics layers, right? And then you have a combined enrichment score that somehow puts together the enrichment of the different layers. And then you can see here, that's depending on the layer that you are looking at, then you have enriched things or not, enriched pathways or not, and some are kind of enriched at every single layer, right? So they are heavily regulated in your experiment. For example, here the biosynthesis of amino acids is kind of regulated at all levels that we can analyze. Then you can visualize this. This is mapped to the pathways, and then this is basically what you get. So this is the pathway with the different time points, and you can see here the names. This is a very kind of broad representation, but for example, here you will have the information for gene expression, but then you can also look at for each individual gene, you can see what is happening at the level. For example, here we have gene expression, methylation, microRNA, and ChIP-seq. In some cases, we have information, in some others not, but you can see, for example, in this case, I think that there is a correlation between gene expression and ChIP-seq data. In this case, I think it was the binding of particular transcription factor, but in some other cases, it's the opposite result. That's indicating that probably this transcription factor is a kind of a repressor of the, or in this case, we have a microRNA that may be having a repressory effect on the gene. And for each of the omics layer, we can also see which are the trends in that one particular pathway across time, right? So here you have an overview, and you can explore each individual node of the pathway, what it's doing across time, and then visualize this for every single omics. You can just change that. One thing that we observed it was that metabolomics are difficult to analyze by enrichment methods. In general, we have like a little enrichment. So we have come up with some other analysis that we call especially like hub analysis. So basically, the idea that we have here is that when we have a metabolite that is differentially abundant, we want to identify we can map and we can see which are genes that are also significant in the pathway neighborhood of that one metabolite, and identify possible hotspots of regulation for metabolites. So basically, it's indicating metabolites that are highly regulated or have an enrichment of regulatory elements in the pathway setting, right? And then we can have some statistical analysis to identify those kind of metabolites hubs. So for example, and we have also, a metabolite class enrichment analysis in which we, instead of doing enrichment of pathways of metabolites per pathways, we look at particular class types, like for example, if they are amino acids or if they are other type of metabolites. And for example, here we will have this analysis of hub metabolites. I think that this was for spermidine, and that was significantly regulated here. And then you could see how the neighborhood was also differentially. That was a downregulation, and then also the genes around them were heavily downregulated. This is one example in one analysis in Arabidopsis, in which we were looking at the effect of the overexpression of this brasinosteroid receptor that is involved in drug resistance. And in this case, I'm showing you what you can find with the MapMan database, which is for plants. And again, you can see some pathways. In this case, is the chlorismate pathways. I think that's we found that was downregulated, and then was involved in this synthesis of aromatic compounds that are listed there. That is something that is leading this capacity to roots of being more active and more drought resistant. And also this lignin synthesis was upregulated, and then you can see how the genes involved in these pathways are jointly upregulated in conferring also the resistance. And that's it. I think I'm right on time. So basically, this is a kind of tool to help in interpreting multiomics data, and that helps you to lead to the formulation of testable hypotheses. We are supporting plant and animal databases. Ongoing work is include novel algorithms for regulatory inference, and we are also thinking of including large language models to help even more in giving you the interpretation of everything that you are looking at. And just to remind you where to find the tool, and that you have available documentation there. And okay, so that's my PaintOmics 4 presentation.